So we call this a situation where the software is actually building its own hardware. All we did was put in a chemical piece of software, and it led to making this physical structure that has biological activity. But we didn't want to make just a small virus. We wanted to make an entire bacterial chromosome. And there were two aspects, as I said. One, could you boot up the DNA? It was easy with the viral DNA in E. coli, but we didn't think it would be so easy with trying to boot up an entire bacterial genome. So this study in 2007 that we published on booting up and transplanting a genome, I think is actually one of the most important ones our team's ever published, because we actually, by changing the genome in a cell, we completely converted one species into another. And it seems like alchemy or something to many people until you really understand the importance of DNA and the importance of genetics and how life actually works. Because this is so important, I thought I'd walk you through it. So we isolated the DNA from M. mycoides. These are very simple cells with just plasma membranes. We needed to know, for example, were proteins required to do transplantation? Because if we're just making chemical DNA, we needed to know if proteins were involved, so we treated it harshly with proteinases and removed all the proteins. We added a few gene cassettes so we could select for that chromosome, and it would turn cells bright blue if it got activated. And we worked out ways to insert that genome into a related cell, M. capricolum. And we thought about this for a long time. We thought we would have to eliminate the chromosome in the recipient cell before we put in the new one. And we worked on a lot of ways to do that with radiation damage, chemical damage. And finally, after trying a number of things, we decided maybe we don't have to do that. And we could use the enzymatic systems in the cell themselves to do this for us. So we have this very sophisticated movie to show you what we think happened. So we inserted the new chromosome in the cell, and for a brief period of time now we have a capricolum cell with two different chromosomes.